רגע, זה מה שהיה פה?
כן, פשוט שלפתי את הרצים.
הוא לא בא? אז רגע,
כן, פשוט אחרי זה.
רגע, זה השניים שמה שלקחתי.
Hi everyone, welcome. So today we have Professor Rafi Haddad from Bar-Ilan University and the Gonda Multidisciplinary Brain Research Center. Rafi's background is in mathematics and computer science. He has a PhD from Weizmann University with where he worked with David Harel and Noam Sobel on coding in the olfactory valve, auto-localization, and developing electric nose. He then moved to do a postdoc at Harvard University with Naomi Chida, and he also worked with Venki Murphy on temporal coding in olfactory valves. Since 2013, Rafi has a lab in Bar-Ilan University. They focus on the neural circuits of olfaction, and they study how neural responses coded and code stimuli and how they initiate behavior. Today we will hear about oscillation and synchrony in neural activity with specific examples from audio-evoked responses.
Okay, thank you very much for inviting me. We have 1 hour, not right?
Including questions.
Yeah, so probably a lot of time, so feel free to interrupt in the middle and ask questions, so it will be also more discussion. If you don't understand anything, you can stop me. Okay, so the topic of the talk is here, how synchrony improves sensory processing, may improve. And the answer is probably in Hebrew, so if you know Hebrew, you probably understand the answer. But I'll try to walk you through it. It's a walk that we have done in the last 2 to 2 years, something like that. So for people who doesn't know, if you look at the brain and you record, you filter out for different frequency powers, there is one of the rule for the strength of the frequency, which we expect to see, but in the brain you will see something like that. There is a bump around here. So there is some increase in the power of around 40 hertz, and goes up to the 200. And it was observed in for many years, and people have thought that this might have is important in neural processing. So the most important thing is attention. It was noticed that once someone is attended, the power of the gamma oscillation increases. And there's a lot of work in cognitive neuroscience trying to understand how this is related, what is exactly is doing. So, and it also was detected in diseases, and sometimes the gamma power is decreased or changed or aberrant. So people they spend a lot of time trying to understand what is the gamma rhythm and what is exactly is doing. We see there's a lot of papers in this field. And there's also several studies, I just picked a few, who show that gamma is important, right? This is a very old one in an olfactory system from Stopper lab, from Gilles Leram lab, and he canceled the gamma oscillation in the mushroom body in insects, and he showed that it hampers similar order discrimination. But then you can ask, maybe this is just because you messed everything in the mushroom body, you just canceled the activity of the inhibitory neurons, and this is why they don't do well with others. A better experiment, more recent, here they increase the gamma optogenetically using by activating the Pavlovin neurons, interneurons, and this way you can increase the gamma oscillation in the cortex. And they show that there is improvement in detection, and this is what's supposed to be with this. There is this better detection of of salient tactile stimuli. So this is better because you increase the gamma oscillation, and then you do better, not do less. But still, you can say maybe you're activating the PV neurons and you did something else in the system. Maybe this is not gamma. And also, and maybe another study that was published a few years ago, they used this auditory and light stimuli at the gamma rhythm, and they show that at least in mice model of Alzheimer, it reduced the amyloid beta and also increased cognitive abilities of mice, suggesting that maybe if you do it a lot for human, you can improve Alzheimer, reduce the Alzheimer disease symptoms. They open up a company, about that they have a company from this lab. But 2 years ago, I think, Buzaki repeated this experiment and find nothing. So maybe we don't have a company. Okay, so the question I'm trying to understand, to ask in this talk is, does gamma the underlying mechanism or just an epiphenomenon? So you can simulate this to a sport car like this, and you can ask, is this the sport car? How the sport cars achieve this amazing speed and abilities? Is it the engine, or maybe it's the sound of the engine? If you listen to a Maserati, you see the sound of the engine is very beautiful. You also invest in that the sound will be beautiful. So is it the sound of the engine, or is really the engine doing the stuff? Maybe the gamma is just the sound of the engine. This is the analogy that we're trying to say here. So we try to address that. First, let's talk about how gamma oscillations are generated. So gamma oscillations reflect rhythmic firing of inhibitory interneurons. That's the overall understanding. Cortical Pavlovin neurons, PD cells, or olfactory granular cells. When those are active, and when you activate them optogenetically, you can see increase in the gamma power. It's believed that gamma oscillation is involved with interneurons and inhibitory neurons and excited neurons interactions. Somehow they interact together. There are also some models that show that only interneurons can also generate gamma oscillations. So what is the model? Just waving hands. I don't show all the models. You have two neurons. One is excitatory, one is inhibitory. The excitatory neuron, the pyramidal cells fire if they get some input from outside. They fire, they activate the interneurons. The interneurons then inhibit those pyramidal cells, and if those inhibitory neurons goes out with inhibition in a time frame of 20 milliseconds, then you will see a cycle that every 2 minutes, 20 milliseconds, those neurons, the excited neurons will fire. Here's a nice presentation of that I found. Oops, it doesn't work. It's always like this. Do you hear anything? Just a second. That's a nice presentation. Those are 5 metronomes, and you activate them asynchronously in the desynchronized way.
If you start these metronomes at different times, they will never fully sync up with each other. After 5 minutes of continual movement, this is still the case. But if you place these same 5 metronomes on 2 bottles, like this, something different happens. 33 seconds, all 5 metronomes are synchronized. Not one stepping out of line or disrupting the pattern. Now why does this happen? Well, finding the answer involves a warning. The following.
Okay, so the idea is that there is a coupler between the excitatory neurons, in this case the metronomes. Then when each one of them is decoupled, after a few of them are coupled together, then they transfer the information through the coupler to the inhibitory neurons, and they start synchronized together. So this is kind of intuition how we generate, how gamma synchrony is generated in the brain. So how does gamma does it magic? How it improves information processing in the brain? This is the goal of the talk now. So we set several hypotheses. One is synchronized spikes are more efficient than non-synchronized ones. So if you have 2 spikes together, if they come together, then they are more efficient from downstream, in terms of the downstream neuron. So this is to say similar to one person trying to move a rock, and if this one person also try to move the same rock, they all fail. But if they go 2 of them together, they will succeed. So even if you have 3 person trying to move each one of them the rock, only 2 of them together can do the rock, can move the rock. So that's the ביחד ננצח, and you need less power to achieve the task that you cannot achieve when you are desynchronized. The reduced number of spikes due to the increase in inhibition does not harm this process. So we said that synchrony requires inhibition. You have excitatory neurons and inhibitory neurons, they work together. So you increase the inhibition, you increase the synchrony, but when you increase the inhibition, you also reduce the number of spikes. So the another hypothesis, another assumption is that you synchronize them, that's correct, but even though you reduce the number of spikes, like here, you reduce the number of person, you can still achieve better because you are synchronized. So this is the second assumption that you need to show. And third, distributed spike can be synchronized. That's another hypothesis that we have to show, because in the olfactory system, but also in the brain, many spikes occur in different places, and if you want them to synchronize, you have to synchronize neurons that are pretty much spread over the brain. In an olfactory bulb, it's a volume of 1 millimeter or 2 millimeters. Also in the cortex, there is a lot of spikes that you have to synchronize. Somehow, those have to talk together and make sure that you have to make sure that they fire at the same time. Okay, a little bit about the olfactory system. This is a cartoon. This is the olfactory sensory neurons in the epithelium in the nose. Those are the glomeruli where those project information into. And from here, there's this projection neurons called matrices. There are also tufted cells. They go to the cortex. So this information goes from here, to this apical dendrite, and then go to the cortex. And those are also being modulated by several interneurons. Here we have the GCL, the ground cells layer. Those are inhibitory neurons affecting matrices, even 2 matrices or more than that, they are connecting them. And we have also some inhibitory neurons here and many others that they are not plotted here. Ground cells are very common. They are almost 10-100-fold higher than the matrices, than the projecting neurons. And it is believed and have been shown in several studies that ground cells are required to generate both inhibition on the matrices, suppressing the fire rate of those matrices, and also to generate synchronization in the olfactory bulb. Also, we know that if we look at this is the culture imaging of a bulb for one order, a different concentration, you can see the glomeruli is shining. And you can see for this order that this is the distributed code. So several matrices, several glomeruli, several matrices are activated by one order. So all those glomeruli somehow need to be synchronized if gamma is really doing the helping this process. So we try to show how this happened. The work was done by Haddad, very talented PhD student in my lab. And this is a slice in which we see the glomeruli, those are the circles here. And those ground cells here are being infected by a virus, which caused them to express channel rhodopsin in this case. Okay, so let's see what we did. The experimental setup, we're manipulating the gamma synchrony while recording already evoked response in the olfactory valve. So we injected viruses into the ground cells layer. This is a GAD2 mice, which means that all the inhibitory neurons have a Cree-dependent expression, so only the inhibitory neurons will express channel rhodopsin. And since we inject into the middle of the bulb, we expect that mostly will be ground cells. Not all, but mostly. So this is the slice again. And now what we do, we record either in the olfactory bulb, or then I will show you also recording downstream in the piriform cortex. We record the perspiration, the spikes, the raw LFP to see all the local field potential, to see all the frequencies. And this is for an example of an odor response. You can see there is a strong power at the 62 gamma wise frequency. Okay. So the first thing we did, we activated the GC, the ganglion cells, with light, that after we express channel rhodopsin. And what we see is what we expect most of all. This is a response to an odor, 0 is the odor, you can see the black, that's the odor. And when we activated it with light, now we're activating the GC, we're activating inhibitory neurons, the response of the mitral cells goes down, as expected. That's a validation of the system. Then we look at the oscillation. So here, this is the spikes at different cycles of the gamma cycle. We look at the gamma cycle at the LFP, and we do rasters of the spikes relative to the gamma cycle. Now what we can see here is they are kind of organized. You can see around here, another 2 columns. But when we activate the channel rhodopsin, when we activate the GC with that express channel rhodopsin with light, it's more organized. And you can see it here quantitatively, and when we quantify all the neurons, we can see that there is an increase in spiked field coherence, which is a measurement of how the spikes are aligned to the gamma cycle, and we see there is an increase. So these slides show that we have a method in which we can increase the gamma in the olfactory bulb, but at the same time we also reduce the spikes in the olfactory bulb.
So each line here is a different neuron?
No, this is different spikes. This is one neuron, just cycles of gamma.
It's one neuron.
Yeah, one neuron.
And what is the pattern?
So it's not about.
So this is one neuron, but this is all the neurons together when we quantify this. Yes?
And what is the pattern of stimulation? So it's just like a continuous.
Yeah, it's a continuous light for all the duration of the odor. Okay. So we did also the reverse. Now we express in the ganglion cells halorhodopsin, which means that we can suppress it with yellow light, with green light. And once we do that, here this is a population, it's hard to see, but the firing rate actually goes up because we inhibit, we shut down the ganglion cells. No, we didn't shut down, we just reduce their activity. Then mitral cells are disinhibited and they will fire more. This is only slightly because the ganglion cells, especially in an anesthetized case, we did this also anesthetized in a way, but also especially in an anesthetized case, but also in a way, they are not very much affected by the ganglion cells inhibition. I will show it later on.
So this is subtracting the spontaneous or the no spontaneous activity?
I don't remember that, but it's
because
evoked.
you
It's
probably
evoked,
raise
yes.
auto spontaneous.
That's an evoked. That's reducing the spontaneous activity, yes. Evoked, when I write evoked, it means that we subtracted the baseline activity. This is why we see the minus here. And here you see that also this is the quantification of the locking to the gamma rhythm. We see a decrease. Those are 2 different measures. This is the vector strength and this is spiked field coherence, 2 different measures. I won't go into the way how we compute them. They're very common. So we have 2 tools. We can increase the ganglion cells activity, reduce the mitral cells, and increase the gamma, or reduce the ganglion cells activity by optogenetically. This causes an increase in mitral cell firing rate, but reduce the gamma. So we have the 2 directions. I want to see what happens, how those two things that we control affect odor processing. So I think the summary is here. We manipulate the level of upstream spike gamma synchrony. Upstream means mitral in the olfactory bulb via GC activation or GC suppression. We tested now how those changes affect downstream odor evoked response. Now we look at the piriform cortex. Yes.
I'm sorry. Synchrony usually means between neurons, but you didn't show.
No, it's not between neurons yet. It is spike relative to the LFP. This is a synchrony. When I say synchrony, I mean the spike relative to the LFP gamma cycle. So we filter the LFP, look at the gamma cycle, and look where each spike happens, and what is the phase in this cycle. So in this case, we are manipulating the activity of the mitral cell through the ganglion cells. What do we record now in piriform cortex? And let's see what we got. Now this is a surprise. When we give odor, this is the response, the black one. when we shine light on the olfactory bulb, on the granule cells, which actually reduce the activity of the mitral cells, right? I showed you before, when I activate the granule cells, the mitral cells, the input to the piriform cortex should go down because there are less firing in the mitral cell. Still, the response increases. That's a surprising thing. So we saw that and said, okay, maybe that's what I'm trying to convince you, ביחד ננצח. The increase in synchrony caused the spikes to be more effective. Although there are less spikes, they are more effective, and we see an increase in the firing rate. And we did this is analysis of the population analysis. We can see here, it's hard to see with the light here, but we can see this, I call it enhance, not increase, enhance, because neurons that fire, piriform cortex neurons that fire to other with excitatory response were more excitatory when we activated the granule cells. And neurons that were inhibitory were more inhibitory. So it's in both ways. It enhanced the response. What happens when we suppress? Mostly the same, the opposite. So this is the other, that's the response for the other. When we activate, when we suppress the granule cells with AuchT, that express AuchT, more spikes should come out, right? Because the mitral cells are less inhibited, we should expect higher firing rate, but we got lower. So it's important that we have those granule cells. We don't want to shut them down. Actually, increasing their activity somehow increase the way information goes into the cortex. And this is the population analysis, same thing. This goes down here. We didn't have much of inhibitory neurons, so we cannot say that in here. So we did another experiment just to understand what is the scope of this, because as I told you, when we activate the granule cells, we're actually reducing the number of spikes. So what happens if we increase the light strength more? There will be less spikes in the mitral cells up to a certain level when mitral cells won't fire at all, right? And then we would expect to see nothing at the piriform cortex. So this is what we did. We changed the light intensity, and what we see here is that the mitral cells goes down as expected, but the fire rate of the piriform cortex goes up, it goes up, and then goes down. So there is a limit for this effect. And the opposite happens here. It doesn't affect much the mitral cells firing rate when we shut down the granule cells, when we when we inhibit the granule cells, because they don't fire much in the first place, especially in anesthetized. But still we see a decrease in firing rate of piriform cortex.
So there's an implicit assumption, at least in the title, that higher firing rate is better, or is it just you mean beneficial in the sense of getting a higher firing rate?
Oh, that's a good question. I'll answer that in the next slides. One of the next slides, I don't remember which one. Okay, several controls that we did. We wanted to understand, is it really the GABA's thing that happens here, or maybe it's just, if I just inhibit the mitral cells a little bit, piriform cortex will start firing wonderfully. Maybe this is not the granule cells stuff. Let's try to inhibit the mitral cells directly. So this is what we did here. We express halorhodopsin in the mitral cells, inhibit them directly, and then what happens is piriform cortex goes down. Piriform cortex needs the mitral cell spikes, obviously, but it also needs the GC to reduce their activity. If you reduce the activity directly, it won't work. Another question we ask, is it the GC, is it the granule cells, or maybe some other inhibitor neuron? If we activate some other inhibitor neuron in the olfactory bulb, which reduce the mitral cell activity, maybe we'll get the same effect. And this is what we did here. It doesn't. We activated the PSGs. This is another inhibitor that is located at the glomeruli layer. And what we see again, mitral cells goes down, but also firing rate of the cortex goes down. So you need a GC. You need to reduce the activity of the mitral cells through the granule cells. That's the only way it works. It's the only way from the ways that we tested, which is also related to gamma, as I showed you before. So let's summarize. We did opposite exhibition of granule cells. It caused an increase in gamma synchronization, spikes relative to the gamma cycle. It reduced the mitral cells activity, but still, surprise, we get a stronger response in the piriform cortex. When we did the opposite, we get the opposite. It goes down, the synchronization goes down, mitral cells a little bit goes up, but piriform cortex is not up at all. When we do the two controls, we inhibit the mitral cells directly, then the mitral cells goes down. The synchrony also goes down for some reason, and probably because we reduce the activity, and piriform cortex go down, and we did juxtacellular neurons, inhibitor near the glomeruli, there are no change in power, there is a reduction in the mitral cell activity, and also there is a reduction in piriform cortex. Conclusion, gamma and not firing rate change is the underlying mechanism. That's what we're trying to say. And that's the question you're asking. How changes in matrices by gamma synchronous affect peripheral cortex and some other representation? It doesn't say that if I increase the firing rate, it's actually improved the information transmission, right? Maybe I also increase the variance, and there's a mess in the peripheral cortex. So we did the following experiment. We took 6 orders and recorded in peripheral cortex while we activated grounds or shut down the grounds. And so this is a response for 6 order, for example. Those are different sniffs. So the order started 0. Those are the two sniffs before, two respiration before, 3 respiration before the order, and those are 3 respiration after the order. You can see here, most of all, hopefully a little bit here there is a response to this order. The 4th one, the 5th one, here's the 3rd one. And we ask how this representation change when we activate the gamma cells or inhibit them. And what we get is something like that. This is the order in 3 dimensions when we plot it on the PCA space. You can see each ball here is 1 order, and you see a lot of variability, and they're very close to each other. And when you activate the channel rhodopsin, you get them a much smaller and more separated. This is visual, and so we did analysis also of decoding analysis. We try to decode the identity of an order, giving all the samples that we had before. If we have activation of GC or suppression of GC, and you can see here, this is correlation. You can see this correlation is also improved, but decoding improved when we activate the GCs or degrade when we suppress the GCs. So we need to activate the ground cells in order to improve its improved firing rate of other responses in peripheral cortex, and also improve representation in peripheral cortex, really helping information transmission from the bulb to the cortex.
Yeah, in the PCA, there was a greater separation, and is it in the direction that is consistent with the representation before the increase in synchrony, or is it totally new representation that's just has greater difference between the groups?
I have no idea.
The PCA, when it was calculated, the eigenbasis was are you aware if it was the same basis as was calculated for the data before the manipulation, or if it was recalculated?
No, it's recalculated. I guess. I didn't do that myself, but I guess that's what he has done. Okay, so this is just to give you an anecdote. There is several papers in the field from 2010 and from 2015, 2018 that show that if you activate ground cells, they did only behavior, and they show that if you activate the ground cells or degrade them, then the mouse performance increases. So we're suggesting that this is the mechanism. If you're activating the ground cells, you're actually improving the information transform to the cortex because gamma is increasing and then firing rate increases in the cortex.
Wait, I'm sorry. This is, the x-axis is the light on. So without light on, it's chanceless.
No, this is correct response. So the animal need to differentiate between 2 orders. If I remember correctly, this is the paper from Gershman. And they show that they learned to discriminate 80%. It takes them around 500 tries. And then if you...
So that's a number of tries.
Yeah, 500 tries here. So if you, but when you activate the light, then they do this faster and also reach probably a little bit more accuracy faster.
When you say more information, just do you mean better signal to noise?
In that sense, yes. I mean, your representation of others is better in the different models. So this way, you can also discriminate better between 2 others. So how downstream neurons benefit from increased gamma synchrony of upstream neurons? Downstream neurons are more sensitive to spikes driving a gamma gradient. That's what we're trying to say. And increased locking increase the probability of distant NTC to fire together. So in the bulb, what I showed is that when you activate the ground cells, you get more spikes locked to the gamma rhythm, the LFP gamma rhythm, which means that you expect to see more spikes firing together at the same time. So this is a study that was done, pre-old study, recording mitral cells response to odor, and they show that indeed when you activate, when you give orders, there are more spikes happening together in the mitral cell, of mitral cells. So you have more spikes happening together at the same time because of the gamma cycle, and then you will get peripheral cortex to respond better because, as in the rocks, you have several spiking that you can threshold, you can change, you can break the threshold, causing the peripheral cortex to fire. Okay, so this is.
So that is only true if the gamma is synchronized between the olfactory bulb and the peripheral cortex, right?
No, you don't have to do that, but you have to all the gamma rhythms within the bud have to be synchronized. That's what this is a requirement. That's what I thought you were asking. If I had measures,
the peripheral cortex also, the cell, the troughs of the gamma should be together so that
yeah,
the recipient cell can respond to this.
If it is oscillating too, then the gamma will have to be in sync. I guess you're right.
Right.
In terms, yeah. But even if it's not, even if it's a trough, if it's in the trough, and then you send more spikes at the same time when it is on the trough, you will get, you have a more chance to fire, to kick it out, kick it and make it fire.
Yeah, if the troughs of the two coincide, right?
I'm trying to say maybe it doesn't. You don't have to. I mean, it's better, but it doesn't have to be. Even if they are not synchronized, and I don't know if they are synchronized or not. I don't remember right now. I think it's debated. Even if they are not synchronized, assuming that you have more spikes happening together in the mitral cells, in the olfactory bulb, then even if they arrive at the trough of the peripheral cortex neuron, they have more chance to make this neuron cross the threshold, regardless of where it is on the cycle.
So maybe it's related. I don't understand. Is there any experimental evidence for gamma at the peripheral cortex?
There is, but I don't recall exactly what. It's debated. Some people say it's more beta. Some people say more gamma. So kind of I have a black hole in this.
What you showed at the first slide in your talk, I think, was visual cortex.
You're talking about visual cortex or peripheral cortex?
No, I'm just saying.
In visual cortex, there is gamma.
But
in peripheral cortex, it's more beta. Okay. So, chapter 1, summary: increasing spike synchrony enhances downstream neuron responses and stimulus representation, despite the reduction in fire rate of output neurons caused by the increased inhibition, and that's what we have here. It's 2 spikes because we're reducing the number of spikes. Each person is 1 spike, but if they are working together, so they can achieve the work. So I'm repeating myself. Synchronized spikes are more efficient than non-synchronized one. The reduced numbers of spikes due to the increased inhibition does not harm this process. And the last assumption that I said in the beginning is distributing spikes can be synchronized. So mitral cells, as I showed you before, I think I brought it here. Yeah. I showed you before that there's an order activates lots of glomeruli that are very much distributed on the olfactory bulb. So you have to synchronize all the mitral cells that fires from different regions at the same gamma synchrony. So how you can do that? So that's something I didn't show you, but it's kind of an assumption that you need in order for this thing to work properly. So that's the second chapter. In other words, this was published before. This study was already published in 2022, I think, by INSA reports, and this one was just published in eLife. So the hypothesis that we set here is this: GC activation synchronize only other-activated MTCs. We don't want to synchronize all the matrices in the body, right? Because they're all firing. They have quite a substantial baseline activity. And if you synchronize all the matrices, then you get a mess. You want only to synchronize the matrices that responded to the other. So in that sense, you would like to have a mechanism which will synchronize the other activated, should be activity dependent. Only those that are active will be synchronized. And it should be also lateral synchronization. You want to combine all matrices that are waiting. This one should be distance independent. I want to synchronize almost all the matrices in the body.
I don't understand. If they're not active, then there is no synchronization.
But it's still fire, so that's going to generate you a lot of mess. You have many spikes all over. Sometimes you get them at the same time, sometimes not. And when you have many, only the others that those are active, you want them to affect the peripheral cortex.
Spontaneous activity.
Yeah. So you're saying maybe this doesn't matter?
So a mechanism that will differentiate between spontaneous activity and older form.
Yeah. But you're raising a good question that I didn't think about. Maybe I'll think about it later. Okay. The network of synchronized MTC is shaped during learning, passive or active. This is another assumption. So if only when there is an order, only the matrices that were activated by this order are synchronized, somehow something like this, you need a network for that, right? You need them to could be somehow connected with probably ground cells, but then you have different orders, new orders, how do you generate this synchronization for every order that you encounter in your life? So the assumption is that you probably this kind of a learning process.
So, but you are trying to find the mechanism for something that it's not clear that it's at all a problem, right? Even if you have gamma synchrony across all neurons, and this is the effect downstream is only rate dependent, so such that only the activated cells will be active. I mean, you can still have synchrony between neighboring cells, even if they don't belong to the same order. I mean, if gamma synchrony is everywhere, and it's also a possibility. Did you find that only neurons that respond to the same order are synchronized, and those that are not those that are less active are not synchronized?
Let's think about it like this. I think it's related to the high question. You give an order, and then you have a microcell here that is firing it at a gamma rhythm, and then this one also firing to a gamma rhythm, and this is not the same rhythm.
Yeah, but this one fires a lot, and this one...
No, let's say the both of them fires a lot to the other, but this one is locked to this is phase in the gamma rhythm, and this one is locked to a different phase in the gamma rhythm.
No, I'm saying that all the cells are locked to the same phase,
You're talking about...
even though they are not related to the same order.
Again?
If you have cells, all the cells in the network are... Gamma is just global. It's
If
not
it is.
order specific. If it's not order specific...
Gamma, nobody showed that gamma is global. That's an assumption that you're making that nobody
Well,
showed
you're
that.
making assumptions that it is specific.
It's specific, each microcell is locked to its gamma rhythm. The specific gamma rhythm within its local neighborhood.
Yeah, but
And another one is locked to a different gamma rhythm. It could be a different phase. And this gamma rhythm, also in this one gamma rhythm, doesn't necessarily have to be aligned. So somehow you need to align those guys.
They can be not aligned or can be aligned. Is there any evidence that shows that the alignment only occurs between microcells that are order specific?
That's what I'm trying to show. That's the purpose of this work, to show that when two neurons are active together, they are more aligned, they're more synchronized.
Only if they relate to the same order.
They're active together, they're
When they're active together, it doesn't have any order that are activate two microcells, there is a more chance that they will be synchronized. Yes, Chaim?
I still don't understand. Is there a global synchronization of gamma or not?
Again, this is debated. I mean, there is a paper right now from Shepherd Lab that suggested that there is a synchronization of gamma, or so there is a synchronization across all the body.
And you assume that it's not?
I assumed, I didn't know that at the time it was published. It was published like half a year
But what
ago.
is the assumption here, that it's not?
Even if it is,
global, then what?
no, even if it is, you have to somehow align the two guys. I mean, you have one matricel here, one matricel here. Each one of them fires a different
You have
phase.
lateral inhibition globally across the...
But then if each one of them is locked to a different phase, then they will not be aligned.
It's not. This is the synchronization. It's not different phases. If it's global, then it's global.
There's a global clock of LFP.
All of them are locked to the clock.
But not all of them are locked to the same phase.
What?
Who said that all of them are locked to the same phase?
Okay, but if this is the case, then there is no question, right? There is a
Yeah.
global inhibition, lateral inhibition that lock all of them, phase lock all of them
Yeah.
to one clock.
Yeah.
Then it's done.
Yeah.
So you assume that this is not the
Yeah,
case.
there was no proof of that yet.
I see. So despite the fact that in your assumption there is no global clock, only local clocks, somehow they're synchronized if they respond to the same
Yeah.
stimulus.
Yeah.
Although they have their own clock.
What I will try to show you is that when both of them are active, they tend to synchronize each other.
So the clock synchronized or not?
It's a kind of, I don't know if the clock itself synchronized, the spike synchronized. Maybe it just changed the phase. One of them was firing at the trough, one of them was firing at the
It's
peak.
hard to lock, phase lock, two neurons which at the same time are locked to their own clocks which are not synchronized. Very hard, right?
Each one of them is synchronized to, let's say there is a global lock. And which,
Okay.
if there is a global lock, as you suggested, maybe it is the truth. then one neuron could be locked to one phase and the other to another phase. Then it won't be good enough. Okay. So this is what we did here. We used a projector and a Tibet-Cree mice. It means that we can express channel dopsin in matrices, and we can activate the matrices directly. And then we try to understand, we have two matrices, and we're trying to understand what is the direct interaction between matrices. That's the goal of this study. And we did this with this kind of activation. We are activating lots of patterns, so we can reveal both the excitatory response, because each time we're hitting, the light hits the excitatory neuron, the one that we are recording from here, then it will fire. And once we are hitting neurons like here, that inhibit this neuron, we will see inhibition also. And we also did several other, and I will show them later on when we reach them. So this is the result of this process. This is a spike-triggered average analysis. We are presenting lots of images with different spot sites, and we're averaging all the spikes, multiplying the spikes by this image, averaging everything together, and we get, where we're recording, there is excitation, that's excitation, and also a little bit of inhibition around it. So this method allows us to see both excitation and inhibition in the factory level. And what we see is that inhibition is quite sparse. There's only one or two around the glomeruli. That's one finding. And when we analyze this in terms of firing rate, we see that the activity change of the matrices that we record is activity dependent. Then the inhibition is strongest when the neurons are firing between 100 and 100 hertz. So this is lateral suppression, lateral inhibition, coming from one matrices to another, which is activity dependent. That's one finding. This is not synchronization yet, this is inhibition. Inhibition is also activity dependent. So only matrices that are active together will mostly inhibit each other, will mostly affect each other. Why this is good, I don't know exactly. Actually, this was shown in 2008 by intracellular coding, an in vitro study, which is surprising that we got almost the same graph. This is a Rafi paper. They suggested that it can increase signal to noise ratio. I don't know exactly how, but maybe we can talk about it later with Chaim. You have anything?
No, I mean, there's a work on efficient coding. When you have inhibition.
What?
Efficient coding.
Yeah.
Through tight balance. That's basically what it does.
So why if it's efficient, but why is increasing the signal to noise if I only apply inhibition between active neurons?
No, the neurons that are tuned to the same thing, one of them will be active and then will suppress it, so they have to be tuned to the same. Well, we can discuss it later.
So this is an analysis of this. We just plot all the excitatory spot in the middle, and this is the inhibitory one, and we can see that there is a radius of inhibition of in the olfactory web. It's a distance dependent up to around 800 microns. And this is the analysis of all the directions. You can see the peak is around 500. So empty to empty, microcell to microcell inhibition is distance dependent and activity dependent. That's one thing that we found.
Do we know if the receptive field or the odor specificity is also organized in a similar way in this area? Kind of like I don't know what it would be called, but like a topographic map.
No, there's no topographic map in the factory. There is a very weak one. That's what we know.
So it's not necessarily inhibiting other odors. It's just inhibiting the neurons that are around it.
Yep.
Like they're not necessarily.
If there's some sense between those, if specific matrices inhibit a specific maybe there is some logic.
You would see
I can't see it. I don't know. Maybe there is some logical structure there, but in our method we cannot read it down. Okay, so now we try to look at. So the suppressive interactions are activity-dependent lateral inhibition, and it's also distance-dependent lateral inhibition. And what I'm trying to show you that in terms of synchronization, it will be also activity-dependent, but it will be distance-independent. Okay. So what we did here, it's a little bit artificial experiment. We took two spots of light. We record from one microcell here, and then we have select another spot. Usually we take either the inhibitory one or just a random one, and we also activate this one. So we do three conditions. One, we shine light on the microcell that we bought from. We expect to see excitation. Second, we shine light only on the lateral one, something that is far away from the microcell, and then we do the pair together. So we try to see what is the interaction between them. So this is an example. I hope you can see that. Those are spikes for different tribes, so each line is one tribe. This is the when we activated this microcell, and then when we activate them together, it's more aligned. You can see more synchrony in those firing rate. This is the quantification. You see that when you activate the lateral one, you don't see anything, doesn't do anything to the firing rate. This is the firing rate. Firing rate doesn't change at all. So we activated one microcell from afar. It doesn't change the microcell, the firing rate of the other microcell, but it's changed the way, the timing of the spiking of this microcell. You can see it here in more spectrogram. You can see this is the hotspot alone, but this is what we get. And when we do it with another microcell far away from it, you get an increase in the power of the gamma rhythm. Now we analyze the distance. So we see an increase as the distance. We see that the change in synchrony. It doesn't matter what the distance is. It's all over the bulb. We have around 1 millimeter and a half that we can check. There's always a change in synchrony, but it doesn't depend on the distance, while it depends on the firing rate. There is an increase when the firing rate is around 20 hertz. So what we're trying to say is activity dependent and distance independent. So two microcells, when they are active, they will interact with each other on spike timing with each other. I didn't show that they are synchronized in the same phase. I didn't show that. But I'm showing that once two microcells are active, they are interacting in such a way that they affect the timing of the other spikes, of the other microcells.
So delta P is the level of synchrony.
Why? I don't remember that.
Because it's negative and positive.
What is P?
It looks like some of them.
This is the delta, yeah. But that's.
Yeah, I'm talking about the delta.
Yeah, I don't remember if this is the gamma rhythm, the gamma locking to it is locking to the gamma, or is it the gamma power? I think it's the locking to the gamma, but I don't remember.
But overall, there is very little change. I mean, it's more, there is a big spread.
There is a quantification here that we did. This is delta. We see an increase overall. It's a small one.
It's
a very small one. It's always a small one. In all those experiments, it's a very small change always.
How tuning it to the specific order?
Oh, we didn't do order. This is just light estimation. So it's activity dependent and distance independent. This is just a mechanism to how synchronize all other activated empty cells in the cloud. It's just we didn't show that directly. Really that. Then we did another just a small study because lots of people are saying that ground cells are doing the inhibition, definitely doing synchronization. We want to check if the ground cells are really doing inhibition. So we studied ground cells to GC interaction. We activated ground cells directly, recorded microcells, and we see no activity change at all. It's mostly doing no change in firing rate. So we're trying to say that the long-standing hypothesis that GCRs are responsible for inhibition in the olfactory bulb, it's not correct. We are not the first to say that. I think we are the first to show that very strongly, I would say. So GCs are more responsible for synchronization microcells without affecting their activity. I know that I showed you before that I reduced the firing rate, but then in that experiment we activated all the GCs. So we shine light on all over the bulb. When we shine light on all over the bulb, we activate all the GCs that we can on the dorsal probe. Here we are only activating a patch of GCs on a different microcell that are located at a distance from it, and we see no effect. Also, interesting finding is that when you investigate interaction between two different microcells, you can see either inhibition, either suppression, or entrainment. So two microcells can have either an entrainment interaction or gamma entrainment interactions or suppression. To summarize, empty-to-empty interaction exerts both inhibition and synchronization. Lateral synchronization is activity dependent and mediated by GCs. Lateral suppression is activity dependent and is not mediated by GC cells, maybe TV, we don't know yet. Investigating this. This increase in synchrony and not changing firing rate enhanced downstream neurons, auto responses, and ensemble representation. So maybe gamma is really the underlying mechanism. Maybe. I think I finished. Yeah.
So, I'm curious. So the mechanism that is driving the coding property of a mitral cell is kind of separate from the gamma entrainment, right? So, if I understand this correctly, so why two cells would respond to the same coder doesn't have anything to do with the gamma.
No.
The gamma just lets them fire in some sort of.
So this is kind of like the bottle in the movie that I showed. There is an other coming, it activates two mitral cells, and because the mitral cells all over the bulb are interconnected by the ground cells, up to 1 millimeter, maybe even 2 millimeters away, through the very long lateral dendrites, then this coupler of these two bottles can make those mitral cells fire together at the same synchrony, in the same gamma rhythm, or gamma phase, such that piriform cortex is happy and fire better and have better representation of others.
So you keep saying about the gamma synchrony, but I wonder whether there is it just synchrony or specific synchrony within gamma? Do you have in other bands no synchrony, or the synchrony is not effective in the same way?
We didn't check that, but my guess will be that it's not synchronized to any other beta or
so
other rhythms.
you don't think there'll be any? It's a kind of a band specific effect.
Yeah, to gamma, which have been shown many times. So I guess Paul B worked for beta and other rhythms. Okay, if I have some more time, I can show you something interesting. New one, do we have? New stuff?
6 minutes.
Okay, that's from the oven, but I wanted to share this again. This is Tal Dalal. I'll show with you this new, I think, exciting results, and then maybe discuss this later with you. Okay, this is a completely different story, completely different project. As you know, cortical regions are organized by sensory cortical regions are organized mostly by some feature of the sensory stimuli, right? In auditory, this is the frequency. In vision, this is the pixel near each other are getting input from close by image points in the image. Everything is organized, well organized in the cortex, but not the olfactory system, right? The olfactory cortex, at least, people believe that doesn't have any order. If you look here in piriform cortex, the projections is all over from the mitral cell. There are many studies, especially from Axel Lab and his students, that there's no organization at all. But why should there be an organization in the cortex? Why should there be an organization in the visual cortex? Why should there be an organization in the auditory cortex? I mean, you can have the same bands all over, spread all over the cortex, and they are interconnected somehow. Why does it have to be ordered? What's the answer? Efficiency, right? Because if they are aligned together and you make some computation near each other, you want to be more efficient. Then put them together, it's more efficient. You save axons length and neuron location. The wiring optimization principle is called. So we did the following experiment. I mean, that was the motivation, to say that there is also an organization in the piriform cortex. We did the same experiment as before. We gave different light patterns each time. This is the light pattern that we gave. It looks like this every time we are activating different mitral cells. So this is the same mouse as before. Channel loops in the mitral cell area. And we have spikes, right? So we can activate those mitral cells. If this is a pattern that activates the piriform cortex, it's believed that piriform cortex is being activated when a pattern of mitral cells is active. So in order to activate the pattern, we did this kind of patterns. We activate lots of mitral cells, hoping that sometimes we will hit those three, and then this piriform cortex will fire. And sometimes we will not get anything, we get zero. And most of the in the time that we get something, We have spikes, we multiply this pattern or image by this number of spikes, it will get more weight, and this way we can reveal which matrices are actually driving the peripheral cortex. So this is what we get. These are 3 examples. This is the raw data, that's the average. We can see hotspot here, hotspot here, hotspot here. And then we are doing all kind of, this is another example. We're doing a z-score, removing everything that is not significant by some kind of a threshold. This is what we get. And then we developed an algorithm which fit glomeruli to each of those hotspots. In terms of, we take the 80% of the data, fit some of the glomeruli, try to guess the 20. If we do guess well, we add the glomeruli, etc., etc. So this is what we get.
These are 3 cells?
These are 3 different cells. So what we show that we can actually reveal the glomeruli. For some, I admit that maybe I missed something here, for example. I didn't cut this one, maybe this is the glomeruli. But more or less we get the glomeruli structure that activate the peripheral cortex neuron. That's nice. Then we can ask, what is the relationship between location of the neurons and their maps? This is what we did here. And I just cutting a very long story short, show you the last figure of this paper that you're trying to write. We are doing this kind of recording either with the poly electrode with 32 electrode contacts with which we know the distance between each of those 2 points, or this one which have 4 shunts, and also we know the distance between each of the contacts. These are several mice, I think 6 or 7 mice, and this is what we get: distance, related, and similarity between the patterns. So we compute the correlation from, let's say, this one and this one, or this one and this one is the same thing. It gives the same result, and we see that there is a very nice correlation between the distance of the neurons in the peripheral cortex and the similarity of the patterns. So what we're trying to say, others are different other examples of correlation, similarity between maps, even from very long distance. We're trying to say that they're interconnected.
This
is just other examples that of neurons that are very distant from each other, which have very nice, very similar maps. And what we see that they are interconnected because they have like cross correlation between them.
But what is the maximum distance?
600. That's what.
Okay, so maybe even 600.
Yeah, I think the conclusion is that there is a similarity.
But there is a bias, then it's not correlation. There is overall bias. It's not uniform.
Say again?
To subtract it. There may be non-uniform activation. The response overall of cortex is not uniform. It's not about a map. It's just overall bias. You have to subtract it.
I'm not sure I understand. Subtract what?
You may have similarity also at the distance of 600.
Yeah, I have.
That has to be subtracted.
Sometimes I have those.
That have to be subtracted. Otherwise, it's not about map. It's not about proximity. A map is about proximity. If there is an overall, look, if the visual scene is not uniformly activating cortex, this part is more than that part. It doesn't have anything to do with the map. That has to be subtracted.
Okay, I'll talk to you later. What exactly I should do. So what you're trying to suggest is something like that. That we have a glomeruli map here, a glomeruli map here, and neurons that are near to each other, they have similar maps. They will collect information from similar glomeruli or overlapping glomeruli or from regions that are close to each other. That doesn't have to be the same. They just need to be close to each other in terms of patterns. And when they are distant, they are collecting information from different glomeruli. Okay, thank you very much.
So what is the reason that previous didn't find it? Richard Axel and company, for instance?
Richard Axel did it with others, with only 3 others in imaging experiment.
This is up to
yeah, this didn't do it. Genetic, and in this 3 other experiment, you can see that. You cannot see that.
Yes.
There is a paper from actually Bob data, but we did it on posterior peripheral cortex. It didn't find much. We took data. I didn't show you.
This is what?
This is anterior peripheral cortex. We took data from Giuliani, which is a student of the Bob data, and he did some recording in peripheral cortex also, anterior peripheral cortex and amygdala. We analyzed the data and we see a similarity between distance, correlation between distance of the neurons and others on the tuning curve. So we can confirm that. It's very weak similarity, but it's significant. Okay, thank you very much. Hi,
בעצם שממש האורגניזיישן של הגדיר, ובעצם מגיעים דברים שקוראים קרובים, יש להם כל דבר קרובים. אבל תהליך של הקרובים, שהם לא תשמעו, זאת אומרת שהאינדיישן הם לא הכי שיש בבית בבית. לא, בקשיבות בין הבאנס, כן, מישהו מהאזורים מסוימים מקבלים בדיוק, אבל לקראת הציבור. אני אומר, תקרא את מה שאתה מקבל במרחקים שזה בעצם אומר, אני לא אומר שהתוצאות לא יהיו. כל אחד המפה הזאת שקיבלת קורלציה. קורלציה ממוצע, כאילו מפה ממוצע. בעצם כמו שאמרת, מרחקים מאוד גדולים. חלק מזה זה בטח בגלל שיש משהו שלא קשור למרחק, קשור לאיזשהו בייס. אני לא אומר שתוצאה אבל שבנתי בעצם מרחקים. בעצם מרחקים. אוקיי, זה מאוד מעניין.
תודה רבה. קודם כל יש happy hour. ומה שאמרתי לפני זה, יש תיאוריות של efficient coding, predictive coding, יש לסופי דנב, קריסטין מקנס.
efficient coding אני מכיר.
אז הרעיון הוא כזה, גם יש שני מאמרים.
הוא נותן זמנים שבהם.
ברמה הבסיסית, אני חושב שנגיד יש כמובן, ואנחנו נסתכל עליהם.
אם הוא נותן אינדקסים, אז בעצם
הם יורים רק כדי לתקן.
שזה הגיוני, כי גם שם גם
את הסטיות, מה שזה
בואי נבדש את זה.
כן, לעשות כאילו תמיד מקורלציה אינפורמציה כדי בדיוק, דווקא המטרה פה להוריד את זה. זה במקור שכל, שהיא מינימום מספר ספייקים נראה בכל מקום. עכשיו, מה שאנחנו רואים שבגלל שיש דיליי, בין באינדיבישן זה גורם לסינכרון. עכשיו, במקרה הקטסטרופיה, הכל מסתנכרן ביחד. עכשיו, קודם כל הקצב של הסינכרון, אני חישבנו את זה לפחות ברייט, זה תלוי בזמן של הדיליי.
אני מסתכל על אינפורמציה שאני רוצה לייצר על ידי הנוירון, על ידי האוכלוסייה של הנוירון. אבל אם אני מסתכל על מה שאני מסתכל על זה, איך הנוירון שמקבל את האינפורמציה מקבוצה של תאים, נכון, זה עדיף לקבל את הספייקים.
נכון, אז קודם כל, הטענה הזאת היא אימון, להגיד שדווקא האוסילציות הן אפיפלו, הן לא הקידוד עצמו, אבל מה שיש לזה ברחבה, כלומר, ברגע שיש דיליי, יש רעש במערכת, אז קידוד אופטימלי מתרחש שיש רמה מסוימת של סינכרון. ואז אפשר למצוא ממש כמה סינכרון צריך, בהתאם בשביל להגיע לאיזשהו קידוד אופטימלי. כן, יש לי שני מעברים, אחד על גייז, אחד על ספייק. גייז זה לא היה לי ספייק.
אני הצבתי אותו. זה היה סימן שלו. כן, כן, כן. בסדר, אני... זה לא בעיה, זה סתם שצריך. תקשיב, בואי נוודא כאילו נתחיל להאמין נראה שהוא באינדקסים של המערך, ואז פשוט נעשה מניפולציה על האקסל. כן, 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 לא, אני לא... וחשבתי להוסיף להסבר בסוף על האיכות של הזה, מה שנקרא... מי שמע היום? זהו, זה דברים שקראתי אתמול. כן, אבל... פשוט שגיפיתי נתן כזה... כן, כן. ממש ניסוחים של... דינמי ניסוח, אבל סרטים אומרים, כן, כן, לא... אפשר עמים, כן, כן.
אומי.
אבל כן, בדיוק משהו...
לא, אני בחדר פה של השערות הסמינר. פשוט... מה? אני בא, אני צריך להגיד לי שאני בוא נפגש איתו, טוב.
אני רואה שגם בגוגל דוקס החליפו את הקונקשנים. משהו אני לא מה עשיתי, זה לא קיצור. אינפורמציה יצאה בסדר, כל הקונקשנים. אז אני אסלט את הקונקשנים ואז אני אסלט.
נראה לי של קנבה. אני לא נמצאתי על מחשב, אבל הייתי לפני 5 דקות, וכן, הייתה לי אפשרות. כן, כן, הייתה העסקה הזאת בפייפל. דרך העסקה הזאת, כן, כן. דרך העסקה פניתי אליהם בבטל.
קושי?
לא, זה היה תרגול קודם, אבל השתמשתי בפרמננט מרקר בשתי שורות. לא יודע מי שם פה פרמננט, אבל ככה מוחקים אותו מתברר, עם מרקר רגיל.
כן, זה טריקים של החטיבה.
לא, אז אני...
פספסת בחטיבה.
הייתי בישיבה בחטיבה. קיצור, למה יש תרגול עכשיו?
לא. סתם... כל הדברים שפה...
לא, העלמתי את כל הראיות, ראיות לפי החומר.
לא צריכה, אני לא משתמשת בלוח, תודה. מה זה? כן. אתה לשיעור שלי?
לא, אני רק תהיתי מתי...
איזה קורס זה עכשיו?
פרט דיגיטלי.
לא שזה.
אני לא רואה את הרקע שלי סתם כי עשיתי נכון, מטושטש. לא, כי האמת שהרקע שלי יפה, אני אוריד את השטוש, אתה יודע מה? סתם. למה? כי אני בכלל באוניברסיטה, אפילו לא בקמפוס שלי. לא בקמפוס, כאילו לא בכלל. האמת שזה יותר יפה ממה שרואים בתמונה. אבל, לא נעשה מה שיש, ואני אומר לך שב-4 יש פה בחדר הזה שיעור, אז שנינו מצוין. אז ככה, אז אני אראה לך, מצוין. אז זהו, אז ראיתי שחימלת פה איזה משהו שמשתף, אני דווקא לשלוח את זה עם זה. לא? מעניין. אני חימלת את זה פה, כן, אוקיי. אז הוא מבקש retrieving the image. זה לא מוזר, כי הרבה פעמים בעצם בוא נראה איך זה הגיע. בגמת העיקרון, פה, אנחנו עובדים על זה כי ממש זה עבד אותו דבר נכון שבדקתי. בוא נראה. זה כן, אבל כאקסס מאוד. בסדר. בסדר. עשינו עבודה טוב. אספת תמונות של התמונה. מה? כי אני הכנסתי את ה... מה? וואו. אני לא שוקר לך. מה אני אספי שבהיה... מה? גרסה של... אני לא יודע, אני שיניתי? אני נראה לי שיניתי. מתי שלחתי לך את הדוקומנט שזה עובד? זה הכל בסדר? רגע, נסתכל. ממש... מה, אז למה אין פה אפילו גרסה של זה? אנחנו במקום, אנחנו בכלל? לסת אותי. נהייתי מעניין. אומר, זה כאילו... זה אמור להיות באקספרט. רגע. איזה בושה, לכן זה לא עובד. רגע. אני יודע, אני כזה אמרתי, אבל ראיתי פה את הזה, כנראה על זה עשיתי את הטסטים. בבקשה, בוא נראה. אם רצים טוב, זה רצו טוב, זה תהליך שאני זוכר. אוקיי, סליחה. אתה צודק, אתה יודע, אז אני כאילו הסתכלתי כל הזמן, לא הבנתי לי שנייה לשם את החיבורים וטסטים, וזה היה אפילו מפעם, ואמרתי, זה לא איך שאני זוכר את זה. אני הייתי עכשיו הולך ומתקן את כל הזה מההתחלה. איזה יופי שאני... בסדר, אז אני מקווה שזה בסדר, אז אתה אומר, אתה לא רואה את זה, אני אגיד לך איפה זה... אתה לא רואה את הקורא את עצמו. זה עולה אצלי, לא, סליחה, ליד. אוקיי, טוב, יש לנו את הכותרת. הנה, אני עכשיו נגיד. נכון, כאילו... לא, כי אני אצלי, זה כאילו... נגיד עכשיו, מה שרציתי עכשיו, סתם מה שרציתי אצלי, כאן
בסדר. חמישי.
כן, חמישי.
נכון?
אני וניצן נשלח לך את כזה שתציצי, כדי שלא נעשה את הליכות בתור הראשונות. אנחנו נראה לי, יש לנו בעיקר לעצב, נסיים את זה כזה מחר, מחרתיים, אני אשלח לנו.
מעולה.
עשינו על וויו בוט.
אוקיי.
בטח את מכירה.
כן. אני לדעתי גם מדבר על הקבוצה הזו של הבוטים.
זה נתונים.
חמוד.
כן. כן?
לא, זה הכי זה. אני רוצה לחשוב כאילו גם כמה הוא מצלחו? הוא מצלחו אחד. רגע. כינו אחד. בעיקרון כאן מחפש את זהו, נסי. נכון. כן, נכון, נכון. יש שניים. אחד, זה מה שזוכר שאמרתי לך שהם צריכים לייצר עוד אפליקציה. אז זהו, זה המקומות. REST API, לא, REST API יש. זה מה ששולח את הדברים האלה. וזה מה שהם עשו בשביל לייצר את זה באפליקציה השנייה. זאת אומרת, זה ה-workflow שלנו באפליקציה השנייה. נכון, את כל הדברים של החומתות שלנו.
איך קודם נקודות?
בעצם אין משהו חוץ מזה. אז למה היה לנו אחד אחר? רגע, זה אקסל שיש. רציתי עכשיו משהו בדיזיין, משהו בזה.
אני לא רוצה שהוא יקבל את זה.
סליחה. זה יחזור גם.
אתה רוצה להכניס 10 דקות
מתוך האפליקציה של טופס מעניות יש חלק מהנתונים, הנתונים הקשיחים שלא קשורים לקומות נשלחים בוובוק, בסדר? והם מגיעים. אז אני אפתח פה את זה, סתם כדי לראות מה אנחנו מקבלים. אז אתה יכול לראות שכך אנחנו מקבלים את הנתונים הקשיחים, בסדר? מידע כדי שמותר, אפילו את התמונות, משום מה נותנים שם ב-REST API, נשלח את זה המקורי, את ה-public URL. השורה החדשה. מחפשים לפי הסאבמישן ID של המקורי, בדיוק, כי יש פה גם סאבמישן של כל קומה. אבל אנחנו בעצם, כי בגלל שאנחנו מקבלים משם הסאבמישן ID שלו, אז אנחנו מחפשים לפי הסאבמישן ID את כל הקומות, עושים להם אגרגציה, ואז כאן, אם אתה זוכר, הוא רוצה את זה, אנחנו שומעים פה, כאילו היינו צריכים לחבר כאילו את ה-A ו-B של כל קומה, והייתי צריך לשמור אותם איפה שם. שמעת אותם פה, ומזינים את כל הקומות, אחר כך יש פה את ההוספה הספציפית של שיבת A ו-B, וכאן אנחנו רוצים לעשות, יש פה איזה מינימום, יש פה מינימום, כי היה מקום שאנחנו רוצים למצוא את ההכרחי קטן, לא, זה מינימום. זה A פלוס B, מינימום. זה יכול להיות גם מקסימום, כן? זה שאלות להפרדה לפני. כן, ואז זה באמת מקורי. עכשיו אני מוחק רגע את כל הדברים, מה זה כקובץ דוקס, בגלל שאמרנו שם שיש סיפור של מה אתה יכול ליצור אותו באקסל, אבל בשביל להכניס את מה צריך לדוקס, אז אנחנו רוצים. סליחה. למה? כאילו, אתה רוצה להגיד שזה ייכנס אחד אחרי השני? כן? חשבתי שאם אני מוחק אותם לזה, זה מאוד בסדר. אוקיי, זה ירוץ ומעיר אחד אחד. הבנתי, הם יכולים לרוץ ממש במקביל. אני חשבתי שתמיד, כאילו, חשבתי שהסקוונט שם פשוט אומר שכזה, תריץ את זה לפי הסדר הגעה, ולא כאילו, אבל תמיד זה חייב להיות אחד אחרי השני. מגניב. מצוין. אני לא חשבתי. אוקיי, מצוין, אז אני חושב שעשו את זה. ועכשיו, יש לנו פה הוספת תמונות של הגימה והפיל, בסדר? אז אני יכול לראות שכאילו, בדיוק זה התמונות של הרשימות, למשל החומה בכלל. יש כמה תמונות הזאת, אז בגלל איך, כאילו, עושה להם ספיט לפי הלאום, ואז עושה להם אינטנציה, ומוסיף אותם, ואז בסוף מוסיף את הבת. כן, זה אומרים. כאילו, כאן אתה צריך להכניס אותו. אימג' עובר. אחר כך יש לנו את התמונות של הקומות. לא, אספת הפתיח כל התמונות, זה פשוט אומר כזה אחרי שאתה מוסיף את כל התמונות, תוסיף כזה פירות להראות, פלוס תמונות של הקומות. ואנשים גם אחרים. שם כאן, אני חושב שהם, אתה שואל אותי אם הם נותנים פה אפשרות, אני חושב שהם נותנים אפשרות של HTML, אבל אני לא זוכר, היו דברים שהם כן HTML. אני יודע שכאילו כשאתה מוסיף פרגרף, אני חושב שכן יש אפשרות. נכון, לא כדאי, אם אפשר אז כדאי לדעתי, פשוט היו דברים שכן אפשר ודברים שאי אפשר, אז כזה אני חושב שאינסרט פרגרף אולי אי אפשר, ואינסרט טקסט, וכאילו... אחר כך זה התמונות, בסדר, אבל בשביל זה היה כל הסיפור, שואל את התמונות, מוסיף על כל תמונה, אין פה כאילו אגרגטור, יש אחד אחד, כל תמונה, מוסיף סליימות של התמונה, אם יש כמה תמונות לתמונה עושה איטרציה, אותו דבר, הקלנה כזה, את התמונה הזו בתוך המספר התמונות הכללי, ונוסיף אותה. אז נתרגם בסוף, היה הרבה תמונות כלליות, ונעלה את זה בתור PDF. Makes sense. הערות, כן. זאת אומרת, יש לנו פה, כאן אתה רואה, זה אתר שיחה, לא, יש פה את הערות תמונה והערות, כן, לא, זאת אומרת, זה לא משוכפל בעצם שתי קומות. בשתי קומות, אז נביא, הנערות כלליות משוכפלות. שואל שאלה טובה, לא נראה לי. הרצתי את זה, תורה של מקרים אחרים, זה שכתוב, כאן זה, אני חושב שהראיתי את זה, ראיתי את זה, אבל לא זה טוב, אבל אני יכול לראות, כן, אני חייבתי. האמת שגם אני צריך, נכון. כן, וואו, איזה מחשב. כן, האמת, אתה יודע מה אני עשיתי? אי אפשר כל פעם, אני חושב שלהם לא כזה נוח מהפילאוט ומשהו כזה. שמעתי את הכל גבול עדיין, ואני פשוט בזמנים פנוי, ונשמע שם. לא, יש שם כזה, אם אין לך, לא, אפילו לא ראיתי, יש שם כזה, אם אתה חסום, אם יש לך מכשיר חסום, משהו כזה, בוא תקח תראה את זה בדרייב. ואז אתה יכול פשוט להוסיף את זה לגבול דרייב שלך. מכל שם, אז לא ננסה עוד טסט, אבל במדי כמה הדברים סחרנו גבולים.